Proteins are one of the four major macromolecules of life. That means that there are three others in addition to proteins. And I can go ahead and write them out right here. So the four major macromolecules of life include, and not in any particular order, lipids, which are fats, carbohydrates, which are sugars, nucleic acids, including things like DNA, RNA, and then proteins. For a little bit more about proteins, we can think about them in the sense of their constituent parts. We learned in the peptides video that a peptide is any time we have two or more amino acids linked together. A dipeptide is when we have two amino acids linked together. A tripeptide is when we have three, and so on and so forth. Proteins are really just peptides, and we typically count something as a protein if it has a significant number of amino acids in the peptide linkage. And significant is a bit subjective, but once we start getting past a small sequence of a few amino acids and start getting into higher levels of protein structure is where we start to consider the peptides to be more of a protein. So what does that really mean? Well, we can take from that there are different levels of protein structure. So level 1 is the primary structure. This is really the sequence of amino acids in our chain. So we can have, for example, alanine, glycine, lysine, aspartate, etc., etc. That's going to be the actual, you can say, letters in our amino acid linkage chain. The next level is secondary structure. And this arises from interactions within the amino acid backbones. And we have several types of secondary structures. So we have, for example, beta pleated sheets, which we can say kind of look like this. And this is just a symbolic representation of what's really happening. So what we see here is, this is basically, we could say, the string of the amino acids we have earlier. Same down here. And then when the different backbones interact with each other through hydrogen bonding, which is represented by these dashed lines, that's when we have beta pleated sheets. Another kind of secondary structure are alpha helices. So they look more like this, where we have the different, something like that. We can just go like that. Here, instead of being a planar sheet, we might have the amino acids spiral into something like this, and then interact through hydrogen bonding in a helix shape. And then there's also less structured ways of having secondary structure, like different coils and loops like that. Our third level of protein structure is tertiary structure. And it's really at tertiary structure. It's really at tertiary and the next level, which is called quaternary structure, that proteins begin to have much more function. So here what's happening is we're getting a lot of beta sheets, and then we're also maybe going to have a loop and then an alpha helix, and then this is all going to come together into one larger thing, which altogether we call the tertiary structure. And it might be represented in a textbook as a sort of amorphous blob shape. And it's not because the individual atoms end up creating this amorphous blob. It's really just a representation of the conglomeration of all the individual amino acids in the chain. So tertiary structure is when we have one unified, very long chain of amino acids that have sections of secondary structure. So just to represent that, here we have our beta pleated sheet, we have an alpha helix, and then we have more loops and coils. That's all going to make the tertiary structure. Quaternary structure arises when we have, let me go ahead and write level 4 quaternary structure. Okay, cool. This kind of structure is going to arise when we have multiple subunits of tertiary structures that combine into a systematic whole. For example, this is a really not super well drawn representation of hemoglobin, which is a protein superstructure that has a quaternary structure because it consists of 4 individual subunits. Each of these is its own tertiary structure. Tertiary, tertiary. But then the whole overall structure is quaternary. So again, proteins are long peptides that have different levels of structure. Primary, secondary, tertiary, quaternary. Not all proteins have a quaternary structure. So many just exist in the tertiary level, but some do. And most of the function really arises in tertiary and quaternary levels. So I can write here. Function arises here.